Okay, so the last class we were looking at what we can learn from structural properties of the brain, right? And then we looked at certain simple nerve systems, and so their wiring properties, and we have arrived at this principle that nerve systems try to minimize the wire length because we asked the fundamental question: what is the brain for? What is nerve system for? And we said it is to put all the parts of the body in rapid contact with each other and enabling the body to respond to the environmental shocks as a whole, right? And for that, you need a rapid nervous system, right? Rapid communication system, which is a nervous system. And one of the things that you need to do to create such a system is to minimize the wire length. We have seen evidence that there is a kind of wire minimization principle in operation, right? In a lot of these examples, you have seen a lot of examples. Now, what we'll do in this section of this talk is to show, I'll try to convince you that this save wire principle or wire minimization principle is an action through evolution right from the beginning. And it is this principle that can explain the evolution of the nervous system and the neuroanatomy. So I'll only present some loose argument because I've been having these ideas for some time, and I also talked about this in my book in the second chapter. But obviously, these have to be, you have to back it up with a lot more detailed studies, both computational and experimental, which I haven't done. But take it as more like a hypothesis. So if you start with very primitive organisms, so let us start with the Hydra. Right, Hydra doesn't have a brain or a spinal cord or anything, just like what humans have. It has a nervous system, but in this nervous system, there's only a bunch of neurons, loosely distributed all over its body. And this is a picture of the Hydra. It looks like a tube with some tentacles sticking out of one end of the tube. And so it has a nervous system, which is called a nerve net, because these are just a bunch of loosely distributed neurons, and they're connected. And they're also connected by synapses. And the signals can go in both directions at the synapse within this nervous system. And Hydra has only one orifice at the top, you can call it the mouth, and also ejects to the same orifice. And it's capable of producing very complex locomotor movements. It moves by performing some kind of somersaults. So in the whole organism, whole body of the organism kind of makes this topsy-turvy movements alternately, and moves from one place to another place. And it does not have any nerve cell bodies, grouping of nerve cell bodies. That is, it doesn't have clusters of neurons. It only has a loose collection of neurons. And such a nerve system is called a nerve net. And a lot of invertebrates have these kinds of nerve nets. Then let's look at jellyfish. Jellyfish also has similar kind of a nerve system. It has what is called a nerve net, which has loosely connected or interconnected bunch of neurons. And this nerve net conducts impulses all over the body of the jellyfish, also integrates stimuli coming from outside and enabling the animal to kind of respond as a whole to the external stimuli. It also has a very interesting sensory apparatuses, like it can sense light using sensors called ocelli, which is interesting because considering that it's a water creature, right? And there is some light that enters, right, even water. So, and that will tell you which way is the sun, which way is the upward direction, in addition to some kind of a gravity sensing, which I'm sure it has. So it has cells which sense light, it has cells which sense balance, and that will enable it to control its direction of motion. Then chemical detection, it has some kind of olfaction, right? It can sense presence of some kind of nutrients in the water and things like that. It has touch sensors. Okay, so this also has a nerve net kind of a nerve system. Now, if you go to slightly higher up in the platform, it also has a nerve system, but this is not a loosely coupled set of neurons. Now, the neurons now start concentrating right into small clusters, and these clusters are called ganglia. So, and you see that in this picture. So you see that these small thicker portions, they're all clusters of neurons called ganglia. Right? And Also, these ganglia in case of a planarium, and a lot of these insects have a very similar kind of an organization nervous system. Even a cockroach has a similar nervous system. And these ganglia are organized in a ladder-like form. So there are two rows, right, hugging along the sides of the body of the animal, and then the two rows which have a chain of each of the rows has a chain of ganglia, and the ganglia at various levels are connected laterally, like you have horizontal connections connecting ganglia at a given level. So the whole thing has an appearance of a ladder, right? And so you see that this is another kind of a template for nervous system which you see as you go higher up in evolution. Then let us look at Eplesia. Eplesia has several ganglia. These are connected by long nerves, and some of these cell bodies are very large, like 1 meter in diameter. Okay, these cells are convenient, and also is a good experimental organism in neuroscience because the cells are large and they are easy to see. You can easily record action potentials and also apply chemicals and look at their responses. Okay, and so this is also a water creature. Then you have C. elegans, which again has a nervous system. Like I said, it has a very small nervous system adherent to neurons. But these also have a chain of ganglia, a bunch of ganglia, clusters of neurons, connected by some bundles of wire. So this is not like a diffuse nerve net, but it is a chains of ganglia kind of nervous system. And same thing even in case of Eplesia. So when you go to octopus, it also has chains of ganglia, right? But among these ganglia, there is the two ganglia at the very top at one end of this nerve system, where the last two ganglia are relatively larger and they start looking like a brain. So for example, if you look at this, if you look at the planarium or platform nervous system, you have these lots of these ganglia. At one end, you will see that there is a fairly large ganglia, two of them, right? And this doesn't look like a brain or anything. But if you go to the level of octopus, right? And octopus is considered to be at the very top of invertebrate hierarchy. So it also has lots of chains of ganglia, but at the very top, the two head ganglia, which kind of fuse, and this has approximately the appearance of a brain with two hemispheres, right? And you can easily guess why it is like that, because, like I said, the body is right a very standard recurrent type of a body plan is like a tube, right? Like I was talking about it even in the beginning. So this tube has some kind of a axis of symmetry, right? If you can cut this on this axis, it looks roughly the same on both sides. And so that is very convenient because you can have legs on both sides, and you can produce locomotor rhythms, and you can move around, right? Now, once body has some kind of a this kind of a bilateral symmetry, right? The nervous system, which is trying to kind of connect various parts of the body, will also mirror the overall body structure. So that will also have some kind of bilateral symmetry. You'll also expect the nervous system to grow into clusters. Right. First of all, why is there a tendency to become into clusters? Because if there is a body, think of some kind of a random body, okay, so abstract mathematical body, organism with some random body, like a rectangle. Sorry. So if I have neurons in this at some various locations, and these neurons are connected in the front to eyes and nose and things like that, these are the anterior connections. And they're also connected to legs and stuff like that, which are at the back. These are the posterior connections. We talked about the anterior posterior connections, right, sometime before, right? Now, if there are more fibers coming from the sensors which are in the front, like I was saying, that more neurons will move towards the front, and so that's why there's a tendency to form a brain-like structure close to the front of the animal, therefore, which will be the head much later. Now, as the number of neurons start growing inside the nervous system, so the nervous system gets bigger and we have more and more neurons. right? So because like I said, our objective is to minimize wire length, right? So therefore the wire among the neurons starts becoming more important than wire between the neurons and the external world, that is the sensors or the sensory apparatus or the motor apparatus, that is legs, right? So the internal wiring becomes more important because there will be more connections internally than connections to the outside, and that's also obviously true as the brain gets bigger, because brain has 100 billion neurons, and among these neurons there are about 10 power 15 connections, whereas number of connections to the external world, that is the sensor, is very small. It is only like order of 10 million or so, right? Which is a much smaller number. That's what happens as the brain gets bigger and bigger. The internal connections become more in number, obviously. So therefore, when you're trying to minimize wire length, since interconnections are more, there is a pressure to bring neurons closer to each other, and that will naturally form clusters. As the nervous system gets bigger, because of this wire minimization principle, or the pressure to minimize wire is so strong, nervous system automatically starts producing clusters of neurons. And therefore, and that's one thing. Second thing, since the body itself has a bilateral symmetry, this central axis, the nervous system also will kind of mirror that symmetry and develop two chains of ganglia which have this kind of simple bilateral symmetry. So you see that. And in case of octopus, octopus has lots of functions. I'll talk about that in a second. And therefore, the head ganglion ends up being more prominent. So that to the point that these two ganglia start fusing, thereby producing what can be described as a precursor to the brain, which you see much later. There's a brain-like head ganglion, and octopus are very smart. They're considered some kind of a genius of invertebrate hierarchy. Einstein's of invertebrate hierarchy. That's how some people describe it, right? And because they can exhibit observational learning. So if one octopus sees another octopus doing something, just by observing it can learn the task. Okay, and so they exhibit playful behavior. So the people have observed octopus doing things like take a water bottle. There is a stream, there's a flow of flowing water. It chucks the water bottle in the water, and then picks it up again, chucks it again, picks it up again, chucks it again. So what is the purpose of that kind of behavior? It's not trying to drink water, it's not trying to throw away a bottle. It's just playing with it. Now, playful behavior is considered a sign of pretty high intelligence, right? And that you see already in an octopus, which is an invertebrate. Can classify simple patterns. It can also predict World Cup soccer. I mean, this is more like a joke, but there is a tradition in, I think it's in Europe, right? In the World Cup soccer, they have an octopus predict the final result or something like that. Apparently, it predicted the result correctly in 2010. But you can take it more like a joke or just a fun tradition. Then if you go to birds, right? Bird brain, although we say that when you say somebody has a bird brain, we take it as an insult. But birds are actually pretty smart. So they can learn to sing, they can teach singing, they can an elder bird can teach a younger bird how to sing. So the teacher bird is called a tutor bird, that's a term. And they can teach a younger bird how to sing. They can even perfect their song. So once they learn the song typically has a bunch of syllables in some sequence. And very often they play with that sequence of syllables. And until they find that song, I don't know, maybe the best, is most pleasant or something like that. So they're capable of playing with the sequence of syllables and perfecting their song. Pigeons could classify over 700 patterns, which is a very large number for a bird. They can also count, so crows up to 3, parrots up to 6, cormorants, another kind of bird, up to 8. And a green heron can throw bread as a bait, right? And wedge-tailed eagle can use stones to break eggs. And there's an example of what is called tool use. That is, if you want to act on an object, right, you use a second object as a tool to act on the first object. Okay, and that is also considered a sign of high intelligence. Because when people first found that kind of behavior in chimpanzees, that was a very big discovery. So So there's a lady called Jane Goodall. I'll talk about that later, right? So that was one of the first discoveries of tool use, and it was a big deal. And that people found even in birds. Now bird brain. So until octopus, we saw there is still chains of ganglia, and there is beginning of some kind of a brain, but it's not really proper brain proper. But when you go to the bird brain, there is actually a brain. It has spinal cord. It has basal ganglia, cerebellum. These are like, so this is these are kinds of things that you already find in a bird. And obviously, if you as you go higher up in evolution ladder, you always find them, right? So this is a radical development in the nervous system structure. But you can see the logic of this development. I will quickly look at that, why. So as you go to other nerve systems, right? Bird. So there is some kind of a brain proper. Cerebellum is also found in lots of very old nerve systems. Like reptiles is a very old species. You will find cerebellum there. The cerebellum is what you see here. Fish has them, right? And fish have them. Birds also have it. Then if you go much higher up to look at mammals, even rats are pretty smart. Rats can learn complex mazes, and rats are excellent, research organism, model organism for studying maze learning, right? Which is used as a standard problem for, because rats learn mazes using a part of the is the one which is important for memory. Because if you damage hippocampus, people have this kind of a memory loss, short-term memory loss. We have seen the movie Ghazni, right, which is where the guy has hippocampus damage. So hippocampus is a very important brain structure for memory operations, right? And in rat, hippocampus to study memory. So rat, right, high capability. Chimpanzees, like I was saying, one of the first organisms animals in which people have observed tool use. So this lady called Jane Goodall, I think this was in late 19th century, not mistaken, or maybe early 20th century. So this lady traveled from UK to Africa to observe the chimpanzees. She discovered, so once she was kind of hiding and observing chimpanzees, and she saw a twig. Right. So the animal pulled out the twig and could eat the ants. Okay, so that's so the animal was using a twig or a piece of stick as a tool to get at the ants. So that was an example of a tool use. Working memory. They could even learn language, and that's one of the few animal species that you can say that are capable of learning language. But they don't have the vocal apparatus to produce speech, like a human-like natural speech. But they could learn sign language. They were, and chimpanzees were taught American Sign Language. This particular chimpanzee called Washoe was able to learn 132 words. And this animal could also teach these words to another baby chimp. And they could also invent new words, right, by combining signs. So language, that's again, we think that only humans have human capability. But even chimpanzees could exhibit linguistic skills, provided you accept sign language as a kind of a language. A human brain. Now, so we have looked at this component placement optimization, right? How nerve systems seem to minimize the wire length, right? And argue that because of this minimization of wire length, with what happened, more of neurons, more and more sensory organs, more diversity in the kinds of sensory organs that you have, right? That's all that you need. And if you just grant that and argue that you can get all these different types of nervous systems simply by varying these conditions. That is, bigger nerve system, more connections, therefore more internal connections than external connections. more sense organs, etc., etc. That's all, right? So one of the things that you see when you go from a rat to humans is in humans the cerebral cortex is much bigger than in a rat. There's another structure called diencephalon. We'll come to it later, which is smaller. Midbrain is also smaller, which is like relative volume, like a fraction of the volume. And spinal cord in humans is much smaller by volume. It's only 2% by volume. The whole. In the rat, it is 35%. So even in animals which are endowed with a brain and a spinal cord proper, there are differences. That is, for mammals, the spinal cord volume is pretty high. It's even bigger than the cortex. Whereas if you go to higher mammal like humans, hello. So the spinal cord volume, relative volume is much smaller than the brain proper. Okay, so if you summarize all these trends that you see in nervous system anatomy, neuroanatomy, we have seen that very primitive organisms like the jellyfish and the hydra simply have a diffuse, what is called a diffuse neuron net, bunch of neurons distributed all over the body. That's all. There's no structure, no brain, no spinal cord, nothing. Now, once the thing is, if you look at the body plan that jellyfish has or hydra has, right? Jellyfish has a tube and then a bunch of tentacles. Jellyfish has a kind of a, I don't know, oblate ellipsoid, more like an ellipsoid. Then you have all this hairy stuff hanging out. Hydra has a tube-like structure with a bunch of large tentacles. The tentacles are also pretty large compared to the body. So it doesn't have any distinct shape. It is not fully, doesn't have the body plan of a tube and a bilateral symmetry. So once you come to that point, like a tube-like structure with bilateral symmetry, right, like leech and cockroach and planarium, lots of insects that have, invertebrates have this kind of structure. And once you come to that point, you have chains of ganglia. And why is that? Like I said, when your nervous system grows bigger, there's a tendency for neurons to come closer because just to minimize wire length. And there's a tendency to nervous system to mirror the symmetries of the body. The body has a bilateral symmetry, nervous system also picks up that kind of symmetry. So therefore, it looks like chains of ganglia. And also, as the nervous system gets bigger and bigger, especially the head ganglions become more dominant, right? Then at the top, you will find like two head ganglion, which can fuse and start becoming some kind of a brain. So that's what you see later. And as that happens, the brain gets bigger, and these fellows, the ganglia start coming together because, like I said, as the nervous system grows, number of neurons grow, there's an increasing tendency towards forming bigger and bigger clusters. So these clusters come together. These clusters also come together. So what happens? This entire thing, right, forms some kind of a nervous system, and this forms a brain. And this tendency, progresses further, and because of that, even this spinal cord distribution, this volume starts entering and starts merging with this brain proper. So therefore, you'll have one large brain and a thin spinal cord. So all these trends can be intuitively anticipated by simply repeatedly applying the Savia principle. But these are only loose arguments. I want to say that, and we need to really back this up with more detailed calculations. So I've asked my, previous years, I've asked one of my TAs to do some simulations, right, on this, yeah. So let us look at that. So let me explain what is happening in this. So you see this in this picture, the two red lines, red bars on both sides are the sides of the body. This is a rectangular body. Okay, I love this rectangular body. And so in the front, the blue dots in the front are the right. Blue dots in the front are the sensory organs. Okay, the red dots are all, you can think of it as motor organs. So, and the black dots are the neurons, and imagine there are fibers going from the blue organs in the front and the red organs in the back or in the sides. And all that I'm trying to do is to minimize wire length, I'm trying to minimize distance. So what we do, we are doing some kind of a clustering, right, of the data. But only thing is, we do something called So scale-based clustering. So we try to cluster neurons at certain scale parameters, right? Then there is a scale in the horizontal direction, there is a scale in the vertical direction. So that is given by these two parameters, sigma x and sigma y. So I just tweak these two parameters and run this algorithm. So neurons are trying to get attracted towards each other. There is black dots attracted towards each other and also attracted towards the blue dots and the red dots. Okay, so the attraction towards blue dots is a lot more because we assume that we have a lot more fibers connecting the neurons to the blue dots, and attraction towards red dots is somewhat weaker. And they're also attracted towards each other. So under these conditions, if you just run the system, see how the dots move. So they stabilize somewhere in the center. They form small clusters which are distributed all over the body, right? So this kind of looks, so for these parameter values, what we got vaguely resembles the diffuse nerve net. Then let us look at one more video like that. Parameters, then that will create increasing tendency towards clustering, both in horizontal and vertical direction. Right. So when you do this, see what is happening. The neurons start producing clusters close to the walls, to the sides. Okay, it's very crude. I mean, I accept it's very crude, but maybe if you do much larger simulations and more detailed calculations, you might get a whole long chains of ganglia, right, hugging both the two walls on both sides. And in the front, you see that it formed kind of two clusters, right, which is kind of like your head ganglia. Now let us look at the third case, where I just change the parameters. Sigma x, that is the horizontal clustering parameter, I increase a lot more. And if we do that, see what happened. All the neurons got clustered in the center, okay, forming some kind of a stem, right, which resembles the spinal cord. And there's a bunch of neurons in the top, which kind of resembles the brain. So this is just a way to present the ideas in a slightly more quantitative form. I'm not saying this is like a proof or anything, but to really establish this theory, you have to actually work it out through a series of PhDs. You need to do a lot of in-depth work. I just want to say, strongly argue that simply by invoking the minimum wire principle, you can rationalize the evolution of neuroanatomy, right? And once you put this on a solid footing, we can ask a lot of much more in-depth questions about the structure of the brain. For example, I keep asking, why do you have two hemispheres? Now, there's some answer to that question because the bicompartmentality, right, of the brain has its origins to the bilateral symmetry of the body. Because we have seen that's how it has come. Right, in octopus, there is a two head ganglia and so on and so forth. But other questions are still unsolved. For example, why is cerebellum outside the brain? And why is the sheet of cells spreading over the entire surface of the brain? Because such a sheet of cells doesn't exist in a diffuse nerve net. It doesn't exist in chains of ganglia. Right? Suddenly, when you go to a higher organism, you find this sheet of cells. I'm not able to explain that using this same Saber principle, but I need to think deeper about it. Right? But it will be interesting to see if you can explain a lot of these features of neuroanatomy by repeatedly invoking this one principle, very fundamental principle of a brain's organization, which is save-wire principle or wire minimization principle. Okay, so with that, we are done with this discussion of structural aspects of the brain. Right now, next I'll go to one more topic. Okay, so now this topic might find a bit strange because so until now we discussed about neuroanatomy, this is the structure of the brain. Right now we will talk about something slightly kind of tangent. I want to actually tell a small story. I mean, I am really fond of this story. So this story comes from ancient India. So there's this Ayurveda guru, right, who teaches Ayurveda to a bunch of students. And in those days, it was Gurukul system where students go and live with the guru, in some forest or something. And they stay with the guru and study, right? The guru taught one of the students, right, a lot about different kinds of herbs that can be with medicinal properties and which can be used in medicine. So the student completed his study. At the end, it was time for him to take the final exam, and he has to be tested. So what was the test? Guru says, give your exam is very simple. You just have to go around the neighboring mountains, right? Look at all the trees and plants in this neighborhood. Get me one plant which does not have any medicinal properties whatsoever. If you can get that, you're done. You pass your test and you can graduate. So the student goes around searching for all sorts of plants, and he has mugged up lots of materia medica. He knows lots of different plants with medicinal properties. He spends a couple of months wandering over the mountains, nearby mountains, and after that he comes back, back to ashram. Because this guy is really disappointed and is like, he was feeling ashamed, like, and he tells the guru, Master, I have failed the exam because. So guru asks, why, what happened? Because I have searched really hard. I could not find even a single plant which doesn't have any medicinal properties. So I failed your exam, I'm sorry. Then the master hugs him and says, no, you have done well, you have passed the exam. Then the student is surprised. What do you mean? How can I? Because you asked me to get some plant, I couldn't get it. He says, there is no plant which doesn't have any medicinal properties. You need to know what are those properties, and you should be smart enough to figure out where it can be used, because there's nothing that is useless. So I like this story a lot because when it comes to understanding brain, understanding how brain works, which is a big problem in neuroscience, there is a mainstream story, a mainstream narrative, which is that right from the days of Raymond E. Cajal, we have learned that neuron is the fundamental unit in the brain. It's all done by a bunch of neurons connected with each other. So we tell the story of what all neurons do. But we forget that in the brain, the other tissues, other kinds of cells, right, which have been there right from the beginning. Now, why do we say that all the smart stuff that brain is doing, all the information processing? Because we use this term, say, information processing, because there's a strong, computer metaphor which we constantly invoke to explain brain's function. Okay, so we say that neurons are doing everything, and whatever else is there in the brain, it's all just is of secondary importance, right? Like that student and who was searching for plants with medicinal value, right? It's like all this other stuff has no medicinal value, no relevance to this understanding of the brain. But point is, if you maybe we shouldn't be too much of a hurry, right, to just go on with the traditional and textbook narrative. Maybe we should take a fresh look at what else is there in the brain before we come to the neurons, and see if there's other systems in the brain which can tell us something about how brain processes information. So in order to do that, let us just kind of become slowly familiar with the brain and look at brain's environment, right? And see if by understanding this environment, you can get any other new insights about brain's computation properties or brain's engines of computation. Now, brain's environment is very interesting. It is a fluid environment. Brain is actually suspended in some kind of a fluid medium. These fluid media are of different kinds. So let us look at the most important ones, most obvious one, which is called the cerebrospinal fluid. So there is this fluid which goes around the brain. I think it's better seen in this picture. So here you can see the brain inside the skull, and you see around the brain there is this liquid, the blue stuff, which is actually circulating, goes around the brain. It goes around the brain, it goes around the cerebellum and spinal cord, and circulates and comes back. And even inside the brain, there are these cavities called ventricles. Like I was saying, the ventricles have been known for millennia, right? Even more recently, like even 500 years ago, people like Leonardo da Vinci and all, when they did their dissections of the brain, they observed ventricles and have documented it most extensive. The ventricles are cavities But in a live brain, there is fluid going around the ventricles. Okay, so that is a kind of a fluid environment in which brain is suspended, and these ventricles have, there are 4 of them. So there are 2 ventricles on the side. They are called the lateral ventricles. So if you kind of want to depict them separately, right? So this and this are the 2 lateral ventricles, and these 2 ventricles combine at some midpoint. So you can think of them as, let me draw a simple picture. So 2 lateral ventricles, and the 2 meet at a midpoint. So this becomes 3rd ventricle, and then from this you go to the 4th ventricle. And after that, it goes around the spinal cord and then it comes back. So fluid flows through them, goes out to the spinal cord and comes back. So this this fluid is called the cerebrospinal fluid, because this fluid covers both the cerebrum, that is the brain proper, and the spinal cord. It is produced right within these ventricles in a structure called the choroid plexus. So they are found. So these 2 are the lateral ventricles, and this is the 3rd ventricle, and this is the 4th ventricle. Right, and inside the walls of these ventricles, so there are these structures called choroid plexus, where the CSF is produced, and from there it goes to what is called the interventricular foramen, also called foramen of Monro. So then the 3rd ventricle and the 4th ventricle are connected using by a pipe called cerebral aqueduct or aqueduct of Sylvius. So that is this one. Okay, this is the aqueduct of Sylvius. From there, it goes into this subarachnoid space through the foramen of Luschka, and there are 2 of these, and the foramen of Magendie. Okay, so again, more pipes. I'm not a big fan of anatomy, I just want to understand the principles, right? And then it goes out and then goes into this space around the spinal cord, goes circulates, and then it goes back into the bloodstream, because the CSF is nothing but, it's almost like the serum in the blood, right? So basically, blood is filtered, right, in these structures, and the serum is taken out, and that is what becomes CSF, and that goes around the brain and spinal cord and absorbed back into the arachnoid villi, right, and so that becomes part of the blood again. So to understand what these terms are, subarachnoid space and all that, we need to look at understand what are called meninges, right? So this is the brain. So this is like a small. What you are seeing here is a small slice of the brain, right? And here, so there are these layers. So this is called the dura mater. Slightly matter here is some kind of matter. It's on a single T. It's not double T. But it has same meaning. It's like some kind of a layer. And then, so this is the brain proper. So this is the, and hugging the surface, there's a thin layer called the pia matter. So you see that, okay, pia stands for soft, right? It's a soft layer, a coating which covers the entire brain. And then there is a dura mater, which is a tougher thing, which is other side. And between these 2, there is the arachnoid villi. So this is called the arachnoid membrane. So within that, you see this small structure. These are called the arachnoid villi. And so the CSF gets absorbed back into the bloodstream in this arachnoid villi. So these 3 are called meninges. That is the dura mater, pia mater, and the arachnoid membrane, which is right here, right? And these 3 together are called the meninges. There's a disease called meningitis, which is an infection of these meninges. So these are the ones that are involved in the disease called meningitis. Okay, so so that tells you an idea of how the CSF goes around. Now, why are we talking about CSF so much, or even ventricles? And what's the importance? Now, right now, we don't think of ventricles much, but if you look at history of neuroscience, right, way back, people used to think that ventricles are very important. because if you look at a dead brain, which is what the ancients had access to primarily, you will just see these cavities, and they thought that, see, right from the beginning, I mean, in the antiquity, people always thought that there is such a thing called soul, right? And that is, there is a separate thing called soul, separate from the body, and that's what gives a sense of being alive, the sense of self, and all that comes from the separate thing called soul. That people just assume that, and then no discussion, and it exists, right? And now the question is, where is the soul sitting? Where is it in the body? Now, for the soul to sit, you need to have some space available, some empty space. Where is such empty space? So people thought that, oh, okay, so within the brain you see these little cavities, emptinesses, right? Maybe soul is sitting there. So many people have thought that soul sits inside the ventricles, right? Or you can call it soul, you can call it vital force, but it sits inside the ventricles. So then, only thing is small variation in terms of the thinking, but people thought that there's soul sitting in brain, or maybe most probably inside the ventricles. Here again, Plato said this vital principle was the brain, and the brain and spinal cord are the coordinates of the vital force. And Strato, another philosopher, placed it inside the fore part, like, of the brain, between the eyebrows. This between the eyebrows business also shows up in Indian thinking, because in our ancients thought there's some kind of a third eye and all that. But when you dig up the brain, you won't find any third eye. But this was the kind of thinking that is there for quite a long time. So Hippocrates thought that this consciousness or soul is in the brain, and Hierophilus, right, made this strange special structure called calamus scriptorius, right? That's where the soul is located. And Erastus is even more weird. He thought the soul is in the cerebellum, because cerebellum, the word basically stands for little brain. Cere is little and bellum is brain. Sorry. Cereb is brain, and cerebellum is like a little brain. And it stated that it is called, it is called silent brain, because even if you remove it, you don't see any major. You can still live, right? So people thought because it is a silent player, maybe that is the location of the soul. Then Galen, like physician, a famous Greek physician, he thought that the fourth ventricle specifically is the seat of the soul. I don't know why, right? How it can be so specific. And Saint Augustine thought soul is in the middle ventricle, that is the third ventricle. Okay, so thing is, so like that, there is a whole tradition of people thinking or believing that ventricles are the seat of the soul. At least from that point of view, we need to take a fresh look at it. I mean, what the hell is going on? Why are people so obsessed about ventricles? Because from a modern point of view, it doesn't make any sense at all. There's a very nice review article by Schiller, right, where he gave a very funny article, a funny title to the article. It said, "The cerebral ventricles from soul to sink." Right? People began by believing that the ventricles are the seat of the soul, but finally now we realize that then there's no soul there. There's just a sink for for CSF. Anyway, so CSF, of course, we don't know. So obviously, we don't think that there is soul sitting in CSF, but CSF has a lot of important functions. So let us look at that. So if you see, CSF first and foremost gives the brain a mechanical protection, because brain is suspended in this fluid volume, and because by density, brain is only slightly higher than water. So therefore, it literally floats in this fluid medium. That's important because brain is made up of very soft tissue. But brain is like a jelly, right? So in fact, that's why in brain surgeries, neurosurgeries, if you want to take out a small part of the brain, let's say there is some tumor or something, you want to take it out. If it's a small tissue, people just kind of suck it out with a pipette. Okay, it's almost like a jelly. It's like a thick fluid. Right. Whereas the kind of brain you might have seen in medical exhibitions, or when you go to a medical school, and right, they'll bring you a brain in a bucket, which is put inside some fluid medium. So that is a dead brain, which is a kind of fixed, right? So it becomes harder because of formaldehyde. It is immersed in formaldehyde. So it looks kind of solid. It kind of looks like the kind of meat that you eat. But the live brain is very soft, almost like a jelly. Now, this jelly-like fluid, when it is suspended in this, so jelly-like brain is suspended in this fluid. Because of buoyancy, the brain, which is normally about 1.4 kilos, will have an effective weight of only about 50 grams. It becomes very light because of this. Okay, so that is important. It is important to have the kind of medium from a purely mechanical reason, because if you take a live brain and just place it on a table, it will slowly collapse because it doesn't. It's not like a hard, stiff structure, right? Unlike a dead brain. So therefore, it's important to keep it floating in this kind of a medium. Then and also it gives protection because suppose you kind of, let's say one day you messed up your exam and you're very frustrated with life and you kind of bang your head on the wall. I mean, I'm not sure if you do that, but let us say you try something like that once. So without this CSF acting like a protecting layer between your skull and the brain, whenever you bang your head, you're going to damage your brain seriously. Okay, so you better not try that, even otherwise. So because of CSF, it has this thick fluid, acts like a kind of protective layer between the skull and the brain. So it'll insulate your brain from the shocks of the world. So next, if you look at another important function is excretion of waste products. Right. So CSF, as it flows around the brain surface, right, picks up potentially harmful metabolites, right, and so removes these substances away from the brain, right, and so keeping it from harm's way. Another thing that CSF does is it acts like an endocrine medium. So there is this system called endocrine system, which releases certain chemicals called hormones, right, and these hormones flow through the CSF, flow along with CSF, and they act on different parts of the brain. So that way it can, so they carry signals, right? So these signals can kind of float along with the CSF and act on different parts of the brain. So hormones released into the CSF from one part of the brain can float along with CSF and act on different part of the brain. So that way you can see that CSF actually participates in brain's communication. So right, so now you can start roping in CSF into the brain's communication network. So you can think of the CSF as some kind of an extended network of communication, where the communication within CSF is through a fluid, right? Communication within the brain is through a bunch of wires, the nerves and nerve fibers and axons and all that. But communication within the CSF is through a fluid. So think of brain talking to itself through an extended fluid medium, which acts as a medium of communication. Okay, so there are these CSF problems that can occur. For example, when the drainage of CSF through the arachnoid villi that gets blocked up for whatever reason, there's a buildup of CSF inside the brain, and that can lead to a swelling of hydrocephalus. So swelling of the brain, which is called hydrocephalus, and which can be caused by the over-secretion of the brain or impaired absorption, of the CSF because of some obstruction in the flow. So this is a brain with hydrocephalus. So look at the ventricles volume. So this is MRI of a brain. You see the volume of the ventricles is so huge. Okay, so normal ventricles are much smaller, and right. So I think let us stop there because now from now discussion gets more intricate. It becomes deeper. So we have just looked at the CSF as a fluid compartment, a fluid medium that surrounds the brain and spinal cord. The other fluid media, which we need to important, are the fluid compartments, which is extracellular compartment, which is space surrounding cells inside the brain. Then the intracellular compartment, which is a space inside the neurons, right? And then finally, there is another important fluid compartment, which is the vascular system, the blood vessels. Now, all these different fluid compartments are in close communication with each other. They're constantly talking to each other, exchanging fluid, exchanging chemicals. And this interaction is very important, right, for the brain to do its job, for neurons to do their job. So you cannot just ignore them as some kind of sidekicks. They are not. I mean, they're playing a very deep, important role, working along with neurons. So let us take a closer look at them and what this fluid compartments are doing, how are they contributing to brain's information processing. And let us look at all that in the next class.